Well, hello everyone, and welcome to the Shoot Apical Meristem Lab in our botany class. We're going to be diving into the differences that we see across different types of flowering plant stems. And today we'll only be focusing on those flowering plant stems, particularly those of eudicots and the monocots. An exploration of other plant stems, such as ferns, will be left for another lab. In today's exploration, we're going to be looking both at the stem as it's developing in longitudinal section, meaning vertically up and down, seeing how it looks like as it grows, that primary growth, the lengthening of the plant, as well as what happens when that primary growth begins to specialize and mature, eventually forming all the different tissue layers that we see in a plant stem. So, first, let's take a look at this image from Wikipedia, where we see a flax stem in cross-section. I want to start here as this is a bit of stem that's kind of difficult to obtain, but very, very important. It shows the development of tissue as the flax stem is growing. This plant is not yet done, so it is not fully mature. But we see all of the elements in this eudicot or dicot. The pith is in the middle, the region indicated with the 1. From there, we see the startings of very thick cell walls, colored in a much darker blue here. These are our protoxylem, our early developing xylem. After that, we have our true xylem of the plant, lots and lots of tubes for water transport. You can see that that makes up a huge portion of this plant stem. And then we go into our phloem, as we will most certainly have. And then supportive fibers, in this case sclerenchyma. Sclerenchyma are not living at functional maturity, comparing to the collenchyma, which are living at functional maturity. And then we have a small layer of cortex. This would be potentially photosynthetic. And then the epidermis. Now, in the lab today, we don't actually ask you to look at a developing plant stem in cross-section, because we're going to look at how this differs in plants that are completely mature. The reason that this maturity difference is important for us today is because we're going to be looking at what these vascular cylinders look like in different monocots and dicots. So let's start by looking at our monocot, maize. So here we have the maize stem cross-section at 40x. It's the first sample that you're looking at in the lab, and there's a lot of reading associated with it because there's a lot of very important features that you need to be able to identify. At 40x, it can be kind of difficult to see all of the space, so we'll first look broadly to make sure that you can identify where the epidermis is, this outside, and then that you can see all of these individual vascular bundles. Now, looking back at our flax plant, we see that there's no breakup in where the xylem is. The xylem is in one continuous ring. That's very different when looking at monocots. In dicots, the continuous ring is a feature. You have concentric circles playing a role in determining the stellar anatomy of all of our dicots. But monocots have individual strands of our vascular bundle placed irregularly throughout the stem. So you do see somewhat of a trend where there's a lot placed on the outside, but there are vascular bundles placed internally to them. This is one of the features that prevents secondary growth in our monocots. If any one of these begin to expand, they'll tear away the others. Whereas, if you have expansion in the stem where it is at least a concentric circle, you can get expansion that would thicken the stem without disrupting the growth elsewhere. So very importantly, our monocots are not going to be showing secondary growth. Now let's go through and label everything that we see. And to do that, we'll have to zoom in at a much higher magnification. So at 100x, we can see a close-up of a singular vascular bundle. Now, the outside of the stem, the epidermis, would be somewhere over here. But the sclerenchymal fibers are visible by their location distal to our xylem. So we can identify our xylem here, big old vessel elements with the corresponding airspace. This, to many people, can look like eyes and a nose. The sclerenchyma fibers are farther away. These are not going to be used to transport water like this tissue, but instead are going to be used for support. So we do see those sclerenchymal fibers supporting our vascular bundle because this is a heavy structure. Water pipes, moving lots of things, dead tissue. It's going to have some weight on the plant. Again, we see the airspace, a region of dead cells, allowing these tissues to breathe. We see some of those primary xylem, the early developing xylem here. And then, of course, our vessel elements, and a little bit of xylem parenchyma, a little bit of living tissue inside these xylem to help regulate things like embolisms, to help general living and repair in this vascular bundle. Because above our xylem, we of course have our phloem, this tissue here that's transporting all of the sugar. We're able to even see the differences between our large companion cells, note above, sieve tube cells, our large phloem cells indicated in teal, so I'm circling that, and then the tiny nucleated companion cells. Big ol' sieve tube cell, small nucleated companion cell. You should see the purple there. So we have our two elements of our phloem here as well. Now outside of that, we see tons and tons of ground tissue, lots of regular parenchyma, all of this performing all of the photosynthesis needed to keep this plant alive. So, in comparing this stem, where we have vascular bundles scattered throughout, to our dicot stem, our ranunculus, buttercup, we see that this, instead of having scattered bundles inside of one another, we have a fairly concentric ring in our ranunculus stem. But just like before, we do see similar features. We see a very distinct epidermis. You even see a stomata here in this slide. So yes, stomatas can, or rather epidermal tissue can still have that stomata, a little bit of breathability. We see the cortex, the layer above, if we're talking about concentric rings, above the vascular bundles, where there's going to be photosynthesis happening. And then we see these support tissues for our vascular bundles. These very, very dark pink-purple sclerenchymal fibers. We then see our primary phloem, the teal stuff on the inside of our sclerenchyma fibers. Again, these are going to be transporting tons of sugar, so they're heavy. They need the sclerenchyma fibers to support them. And then inside of our primary phloem, we get our primary xylem and our large scattered vessel elements. Now, as we go inside of every single eudicot stem, we encounter the pith. The pith is a tissue layer of parenchymal cells that grows on the inside of the xylem. So if we have our xylem here, they're growing on the inside. Now some plants, to lighten the load and grow taller, form a pith pith cavity, an airspace that prevents them from having an intense amount of weight, and instead can grow taller faster without supporting that many cells. We do still have a pith on the inside of our xylem, where you're going to find some parenchymal tissue that's going to be performing photosynthesis. And then a feature that I don't ask you to label, but that you do remember from lectures, may be some of our parenchymal fibers. Now, parenchyma are cells that are living at functional maturity, just like parenchyma, but they are supportive. You may be able to identify some parenchyma fibers near the epidermis with irregular thickening. So they'll be thicker on one side than another, kind of like supportive rebar. Now, just to drive home the fact that all the eudicots are fairly similar, particularly when we're talking about herbaceous growth, we'll take a quick look at a sunflower stem, another stem that will be available to you in the lab itself. Here, just like before, we see an epidermis, we see a cortex, potentially some parenchyma supporting the epidermis with the parenchyma of the cortex below, all of the regularly sized cells, just like we see here in the pith region. And then in sunflower, we see this almost caricature of huge amounts of sclerenchymal fibers with a phloem, protoxylem, and vessel elements or later developing xylem. So this kind of illustrates a little bit better what I like to call the vascular bundle sandwich, where you've got supportive fibers up top towards the epidermis, phloem living in the middle, and then the xylem going from small tubes developed early in growth to large tubes developed later in growth. Our stem in cross section, looking both at monocots and dicots, we're going to take a look at the shoot apical meristem in longitudinal section. Now I'll note here that we are not discussing any shoot apical meristems for monocots, as oh boy, they are complicated. My wife did a fair bit of postdoctoral research on them, and they are not the most simple thing to visualize. So for those of you wondering why we're focusing specifically on dicots like this coleus, it's because they're much easier to follow. But still, it shows a similar structure as you'd expect to see in all flowering plants. So we have a series of images here going from 40x to 400x, looking at the different features in our shoot apical meristem and its surrounding features. So we see the leaf primordia, the developing leaves. We see the protoderm, the early developing dermal tissue. I'll zoom in a little bit to get a better view on it so you can see the protoderm, all of the cells that are going to become the epidermis. And then on the very inside, we see a procambium. And then a ground meristem surrounding that. Ground meristem will become our pith, very in the center, as well as our cortex, because these are into concentric rings of parenchymal tissue. And then our procambium is our vascular tissue. You can see the vascular traces here in the leaves. And then what will eventually become more of our leaf primordia. So we see leaf primordia setting off here. Now I'll take a moment here at 400x to make sure that y'all have the similarities between lecture and lab, where you should be able to identify the tunica corpus. Here is our central zone with our 3 layers, the L1 and L2, the tunica, and then L3, our corpus. Beyond that, we get our meristematic regions where you start differentiating between the procambium and the ground meristem. Now remember that our tunica layers the L1 and L2, the surface layers here, have different directions of cell division compared to our corpus layer. If that doesn't mean a lot to you, please make sure to go back over your notes, as our anticlinal divisions and periclinal divisions, anticlinal meaning opposite the axis of growth, so if we're growing vertically, anticlinal are side to side, and periclinal are with the division of growth, periclinal really being defined in our L3 and anticlinal being defined in our L1 the most. We can see how this tip will actually grow upwards. And this hopefully shows you how our leaves and the vascular tissue associated with the leaves is connected to that of the stem. They mature together in our shoot apical meristem and then differentiate much later once all of the organs are formed, because these, after all, are plant organs. So that will wrap up the microscopy for today's lab, having looked both at our shoot apical meristem in longitudinal section, seeing all the features as they develop, and then in cross-section, in a developing cross-section where we see not everything just yet, in a fully developed cross-section of a monocot where we see the dispersed vascular bundles, and a fully developed cross-section of our dicot where we see the concentric rings. We'll end the lab by talking about how this stem structure relates to plant phyllotaxy. So our discussion of plant phyllotaxy is going to focus on two different modes in which our leaves get spread across the plant. In an alternate phyllotaxy, this cartoon illustrates that we go from one side to the other. And I've shown some images of some of our local flora where we see alternate phyllotaxy. So here we have a stem, and then we have a leaf going off of each side, one after the other on the stem. Same with this more woody plant. We've got leaves going off alternately. Now, if we look at our other type of phyllotaxy, opposite phyllotaxy, we see leaves going off on each side of the stem simultaneously. Like in this mint plant here, we see them going off one side, and then it switches the side, so it'll alternate which side it's going off of, giving the plant kind of a boxy appearance. If they are always going off of one major raceme, one major axis of the stem, we see opposite phyllotaxy here. Now, based on what you know about opposite and alternate phyllotaxy, what do you think this coleus is going to look like in real life? That's the next part of the lab. I'm going to get away from the computer and head over to the greenhouse to take a look at some of the phyllotaxy of the plants in the greenhouse. See you there.
Well, hey everyone. So now we're in the greenhouse to take a look at a couple of our different plants and see if we can assign some phyllotaxy to them. So I obviously want all of you to explore the greenhouse yourselves. We're not going over all of the examples, but I just want to give an illustration of some of our alternate versus opposite phyllotaxy for our plants here. So let's start here with this succulent. Now, you'll notice here, when looking at the flowering stem, that it's very easy to identify that these leaves are on one side, on another, and staggering around the stem. So this is kind of a clockwise pattern as you go up the stem, but definitely alternate. Down below, it's a little bit more difficult because they're all clustered so much. Hopefully y'all can see that. But you still see that they are not letting off on opposite sides of the stem. Now this pattern of climbing leaves is very common. You'll see on this fig here that we've got, again, one side, another side, and it's almost like you're climbing stairs as you go up this fig in terms of alternating leaf patterns. Now we can come over here to our first member of the mint family. In the mints, the Lamiaceae, you have a lot of plants that are doing things in opposite venation. So you've got two branches coming off at the same time. You can look at multiple different locations on this stem, and you will always see opposite branching patterns, whether you're looking vertically, horizontally. The coleus here, which you saw earlier this class as our coleus shoot apical meristem definitely has those opposite venation patterns. We again have another opposite plant here, shooting off like wings on each side of the stem, alternating which side they're letting off of, but again, opposite nonetheless. Coming over here, I certainly recommend that you give this one a smell, rub its leaves. This is a scented geranium. Oh my goodness, that's lovely. And you can see the fact that the stem, the petioles of the leaves are coming off on alternating sides. Again, kind of in that different times around the clock, swirling pattern around the stem. We can come over to see our jade plant. Again, a little bit with the opposite here, like wings on each side, as long as someone hasn't broken them off. And then the last plant that I want to go over is over here, our calocarpa. While it's still got leaves, chances are that if you're doing this lab in the heart of winter, you won't actually see leaves on this plant. It will shed them very shortly here. But where the leaves are, you should be able to see that they're coming off on opposite sides of the stem. So it's very, very distinct that they're both spreading like wings, a little bit of opposite venation. It is a very fragrant plant. I can even smell it through my KN95 mask. It's lovely. So that's it. Have a good time exploring the greenhouse, and I will hopefully see you guys for the next lab.